and welcome back to SARS-CoV-2 program. Let me share the screen. And this is just another reminder that these sessions are recorded and will be available on the forum after the processing of the video and after it's uploaded. So good morning, everyone, once again. Welcome back to the SARS-CoV-2 program, which is an online program for beginners in bioinformatics. And we are going to speak in this program about the analysis of SARS-CoV-2 genomic data. We'll talk about how to find appropriate genomic data for analysis, how to apply some of the analytical tools to analyze big data, and how to read COVID-19 publications with understanding, so that that understanding can help us understand key principles of vaccine and drug design, as well as what's going on with this epidemic and how to filter through the hype, understand what's really going on, and base our decisions on data. So today we're going to be talking about using genomic reference databases and references in general. So we're going to dig a little bit deeper into the mapping of those reads on the reference genome, understanding what a single genome can reveal, what multiple genomes can reveal, and we'll build it up towards the understanding of sequence alignment and quality of alignment. Later on, we'll apply this understanding to the ability to compare different sequences of genomes that are available on in the public domain. So before we jump into that, I wanted to go quickly and review where we are in terms of the program and see how everybody is doing. So let's go. We have two different programs combined here. So we actually have a program for Louisiana Biomedical Research Network. And on the Louisiana Biomedical Research Network, this is the organizational link. You can see that some of the forum postings had session recordings, session files, and resources. And any questions that you might have, this is also where to add those. We also have our common online program, where we also have a forum, the same exact forum that has the recordings and all of your activity. And so if any kind of questions come up from the previous session or overall, this is where you would want to put some of your questions so that we can address them. Okay, the other thing that we can see here is the overall progress as well as the syllabus. So to look at the progress, on the progress, we are going to be looking for those of you that are completing online materials, performing the analysis that we discuss in class, which will result in your own pipelines. And also you'll be able to check how you are doing overall compared to some of the other users on the platform. So I did want to remind you here that we have this point system. Really, the point system is both for you and for us. For us, it's important to understand how well you interact with some of the materials that we offer, and also helps us understand who is able to go through the material in the time frame that we allocate for us to improve the program overall and to understand the different audiences that are interacting with some of our materials. It's also important from your standpoint, because you can see how the material that we do in these sessions is actually complementary to the material that we have online. So Nayati here is saying, I don't understand where to find the material that we need to complete. Yeah, so that's exactly the reason why I'm talking about this. So let me actually go through where all the material is found. So here's the progress tab. So again, this is the link overall to the program. Let me paste that in chat. So that's the link. I would encourage all of you to review some of the materials that we have here. And here is the syllabus. So on the syllabus tab, Niyathi, if you could please also let me know if you found these links. So the syllabus is found right here. We talked about next generation sequencing, what next generation sequencing techniques there are, what are reads, sequences in different file formats. We then continued to apply that understanding to a specific example project that really demonstrates how novel viruses could be identified in a host sample. And so that's what we're continuing to speak about today. So whenever you see the topic, you'll see it right there. And then you can also see some additional materials that you can click on. And some of those, a lot of those are actually based on either what we cover in the sessions or what we cover in these courses. So we try to make some of these materials available for you. You can also find them independently under courses. You can go into the courses or the projects to find some of these things. And on the progress tab, what I wanted to show you is that from the beginning of this program, let's just take it from the beginning of this month, you'll be able to see how different people in the program are utilizing some of these resources. Now, these resources, the courses online, contain lectures essentially that explain different things, but also some assignments. So a lot of you have been completing the introductory courses. Those introductory courses don't have a lot of practical assignments. They're built to introduce you to these topics. But once you get into the more advanced courses and projects, there's a lot of practical assignments that you can start working on. And so here you'll see you know, some of those, for example, Transcriptomics 1, 2, and 3 will have a lot of different assignments that you can complete and understand how some of these theoretical things are actually going to apply in a practical way. So here you can see the activity of this group over time. And it's pretty good. You can see that people started with some introductory materials, eventually started applying those skills to some pipelines. And here you can see kind of overall, there's a few tabs here, but you can see how different people from the top ones all the way to the people that have not really started that much yet themselves. So what do these points represent? These points represent the different types of activities. For example, you get 500 points for completing a course, 100 points for running a pipeline, 100 points for updating your pipeline, and 10 points for group activity. And the goal is to really track and encourage for you to utilize the resources that we've assembled for this program in an effective way. So if anyone has any questions about how to use these, I hope that that was kind of clear and you were able to do some of the materials that we've posted there. So does anyone have any questions about how to find different resources or how to use these resources that we post on the organizational page? And also let me know if you were able to see the previous post that I added after our session on the specific data sets and the reference genome that I pasted on the forum. I know that some of you have because I've seen some of your updates. So maybe if you have found those, put in yes, and that way I'll know. Okay, Andrea said yes. Okay, Kita, Benjamin. Okay, excellent. Yeah, so you'll see some of these things here. And the point was to really point you to some of those materials. I'll just show you, for example, where to find that: session data files and presentations. If anyone wants to have any kind of a reference publication that you saw me reference, or a data set that you couldn't find, I'll be posting them here. And if you have any questions, you're welcome to add here more. Like, for example, Adekunle added here that he ran the pipeline, and I can take a look at that pipeline. So we'll go over that today. And Nayati, no, you, if you're logged in and you're a member, then you should not be asked to pay anything. All of these courses should be included with the project. So if you have any kind of a challenge accessing some of the course materials, please do let us know, and we'll make sure to add you to the group so that you don't have those issues. So the people, the person to contact about any of those issues is Bibsha, and her email is marketing@pine.bio. So if you could please make sure to ask her in advance about any kind of access issues that you have, either to these materials, the courses, the platform, anything like that. So please make sure to reach out to her. Okay, so let's review what is it that we have covered up until now, and what are we trying to address in this session. We talked about different methods of sequencing metagenomic data that includes the microbiome and a subsection of the microbiome that's called the virome. We talked about metagenomic sequencing in general, that there are several different techniques that could be used, from amplification of specific types of organisms to amplification of specific types of genes that can help us identify diversity. And there's a project in Metagenomics 1 that illustrates that process of targeted or amplicon sequencing and how to use it to understand what happens with microbiome changes in humans. So then we also talked about the process of next generation sequencing itself, how the digital data is produced, and what are these individual reads. Here you can see an example of sequencing from a beta coronavirus sample taken from Wuhan, the Wuhan Institute of Virology. And we talked about how there is this assembly of reads aligned to some reference genome, and we can analyze the reads, and we can also take a look at how these reads, what kind of insight they can give us in terms of the reference genome. And then we also talked about the different types of formats. So we talked about FASTQ files. So FASTQ files are those reads that you just saw previously. FASTA files, sequences of nucleotides or amino acids, and the GTF or the GFF transfer format. So the format that represents the positions of different sections in the genomic sequence that we're using for reference. So up until now, I hope that those concepts are really becoming clear. You're able to use some of those. We'll go through some exercises in today's session to make sure that everyone can follow along. Hopefully you've been able to find your account information and everything like that. If you have any specific questions about that, then let's please take a look at those questions now. So are there any specific questions that you had about the previous sessions? Okay, so again, the goal of putting those questions on the forum is to help prepare any kind of specific material that we can review in these sessions. And here's an example. So again, I posted this on the forum and should have come up on the activity tab, where you can see from the previous session, session 2, we use data for visualization. So when we align the reads to the reference genome, we'll check whether any of you have been able to do that, and I'll show you how to do that again. And then also how to identify some of those reads and how to find exactly what you're asking there. Where do we understand what kind of data this is? So before we jump into this, did anyone have a chance to do this practice assignment? If you have tried it, then please type in yes, so I know how many people have been able to try. Okay, so Benjamin has, and Kita. Okay, so we have a few. Okay, Manish has not tried. Okay, I know that Adekunle tried. Okay, yes. Okay, so again, you're gonna have some more time to do this. I think it will really help you understand what can go wrong when you try to do this on your own. If you are able to reproduce it correctly, it will also give you a sense of how to do it next time and apply this to a project of your own. So please take the time. We'll go through these steps again today so that we go them slowly and carefully, and you will be able to see how to do this. So if you want to follow along, please follow along. Okay, so we have this process that we described last time. A biological sample is taken. From that biological, okay, Abilash is saying, I don't have access to these files. So again, these files, have you seen, Abilash, the forum? Let me show you where to find. Okay, so let's go and find, this is the post. Have you seen this post? Oh, okay. You're registered in summer training. Yeah. So that's a problem. That's a problem to discuss with Beepsha. But okay, so here are the files for today's session. Since you're in here, you can reference this. So what do we see here? We see here a link, right? This link will open up as a Dropbox link. You'll see here some files, right? And you can just click on download, direct download, save them to your computer, unpack them, make sure that you have these two files. So that will be important for today's session. So what is the process that we're trying to replicate here? We have a biological sample. The biological sample is, there's RNA or DNA that is extracted and then sequenced. And when we have the sequencing, for example, RNA-seq sequencing, when we measure expression of genes present in the sample, we're typically going to take genes from a reference genome. So for example, if these were human cells, which we'll talk a little bit more about the bioproject and what these data sets are to understand what kind of genome to use and why we're doing what we're doing. So we take some sample, we look for genes expressed from the human genome. We have here the human genome already on the platform. And then once we have those mapped, we want to take the unmapped reads, so reads that did not map on the human genome, because we suspect that they are viral genes. So instead of looking at the human genome, we're going to start looking at the viral reference genome. And here we have the link to the viral reference genome. Okay, a few questions here. Adekunle is saying, I also had challenges viewing the visualization after running the pipeline, hope to revisit. Also, after searching for those specific samples, what are we to do with them? How do we copy the links to use rather than download them? Excellent question. I will address these questions in today's session. Okay, so what did we do first of all with the files that you were provided with? We go to the platform, and on the platform, we are going to use the RNA section, RNA-seq section, because these are gene expression files. And instead of going through the process of first mapping them onto the genome in the human and then finding the unmapped reads, I already gave you the unmapped reads. So just to understand what is in the unmapped reads, you were supposed to do the following. Let me show you. So you download, the direct download onto your computer. Once the files are downloaded, you unzip them. Inside, you have a reference genome and an SVL file. Now, one thing to remember about this SVL file is you have to place it in the right input. So let's go to the platform, and please follow along, and please ask me if you have any questions about what I am doing. So here, as you can see in the name, if you open this file in any text editor, you will see that there's a name of the FASTQ file. So .fq stands for FASTQ. And you will see underline SE because these are going to be single end reads. Why are they single end? Because we're going to take them from the unmapped section and they're converted to single end. No matter what kind of input you originally had, after you have the unmapped reads, they're going to be outputted as single end. So now they're single end files. And instead of using an R model genome GTF, we're going to say R genome, just a reference genome, because we will upload the genome. It's not one of the genomes that's already present on the platform. And we only have a FASTA file. We don't have a GTF file to use. So we're just going to use the R genome. Then we'll go to file upload. And here you can see several different options. You can use add files, add from link, NCBI, or URL bulk upload. Okay, and Keaton is having problems accessing the platform. So please, if you could describe in a little bit more detail by email or on the forum, what are some of the challenges that you're facing? Maybe include some screenshots of what the problem is so we can go in and check for you. Okay, so then I have this button, URL bulk upload. Important not to upload that file anywhere else, but upload it right here. Once you click on URL bulk upload, you have to go to wherever you saved your files and use this SVL link. Okay, so now some of you said that you had problems after going to 100%. So you should be able to see here a button that says organism, because the next thing is to upload the reference genome. How do we do that? We now use upload files. So we go to upload file, and we use that FASTA file that was provided. Okay, and this is for the purpose of making it simple. We already have all of these files accessible. You just need to understand which one to put where. Group selection, you have to see here those 3 files that were described in the SVL, right? So again, take a look right here. You have a path on the server, and inside the path, this is the name of the file. So you should see 3 files as we see right here. Do not need to put them in any group because we're not comparing anything to anything. So you just press on cancel, no groups. And then you only have a couple of steps to add here. So bowtie2G maps all of those reads onto the reference genome. And then visualization. Okay, after you've done that, you click on end, save your pipeline, and run it. Okay, so has anyone been able to successfully visualize this and see the results? Let's take a look at what the results would look like. So if you had successfully done that, please type in yes. Okay, so no one has been able to successfully do this yet. So if you would like to follow along in today's session, I'm sure that pretty soon you will have the results just like I will show you today. Okay, so here, after you have completed this process, your pipeline should be done. It should not take a long time because these are already smaller files. Okay, so here's some interesting problems. So Sravia was able to do this, excellent. Marudula is having trouble to download the SVL links. So Marudula, if you could explain exactly what do you mean by trouble to download the SVL links? Okay, Gandhi was able to do this, great. Joe Bedell is saying my company is blocking the site for the data download. It's a Dropbox link. So we could email you potentially. Which downloads do you mean? Do you mean downloads that I sent? in the link, or it downloads from here? Because down here, you don't have to download anything. Okay, the bit.ly link above is a Dropbox link. So if you have trouble downloading a Dropbox link, we can put it on the website potentially, and you can download it from there. So I'll try to do that again. Let's try and see if you can do that. I also want to, okay, great. So I will do that because Dropbox is supposed to be more accessible, but anyways, we'll try that again. All right, but for those of you that have gotten to this place, you should be able to see this link for visualization. And the visualization link here, you will see that it actually contains, let me find the appropriate one. So here's the result of that visualization. The result of the visualization is going to be fairly heavy because, as you can see, we have the reference. Okay, let's just simplify this really quickly. We have the reference genome, the nucleotides. Then we have here the coverage. So coverage means how many reads are found aligned to this position. How many reads do you see on average aligned in this one sample? Around how many reads do you see aligned? 2,000, exactly. So a lot of reads, right? Compared to, for example, your standard, amplicon sequencing or something like that, this is a lot of reads. And this is across 30,000 nucleotides, right? So it's a fairly long genome. And so you really have thousands and thousands of reads across this whole genome. In fact, if you did get this result and you scroll all the way to the end here, you'll see that there's over 50,000 reads towards the end, okay? And the reason for that is that there's amplification bias in the process of sequencing. And also this is expression of specific portions of the genome, and that last portion of the genome typically has an increased level of alignment to that portion. But here we have at least 3 files. Okay, so one thing that I wanted everybody to be able to see is that you can have a number of different outputs. So actually, not to go through that interactively because it's going to be challenging for all of us to see that at the same time. This is the output that you should have gotten. Again, I only selected here the coverage and not the actual alignment of reads. But let's take a look at what we see here. Okay, so what is your interpretation from what you see? Okay, we have 3 files. Okay, which one does not have that virus present in a sample? Which sample does not have the coronavirus in it? Okay, I'll let everybody answer that question. All right, so most of you correctly identified that the last one does not have many reads that actually line up to this genome. In fact, it has, I would say, close to 0 reads, apart from that last section, which seems to have a lot more reads in all of them, right? So eventually we'll take a look at what is happening here, why so many reads are actually being mapped there. It's not because the virus is actually present. So here we see that in both of these, there is some, right? There's a lot more down here. And even though it seems like there's almost nothing, it is only because this scale is so huge. There's 90,000 reads that map all the way over here, right? So in the middle, right, if we actually scroll in or zoom in, we will see what you are seeing right here. For example, for the second one, there's still over 2,000. So obviously 2,000 from 90,000 is still a big difference. So you're seeing it very close to zero, but there's still a lot of files in there. Now, if you just did the visualization, you should be getting something like this. And you can see that it just has the reference genome because we did not upload a GTF file. How do you upload a GTF file? So here I was able to upload a GFF file. So how do you find a GFF file? Oh, sorry. The way that you find the GFF file is you go to NCBI, and here you can see actually you can see it right here, NCBI SARS-CoV-2 sequences. So for example, if you go to the reference genome, this is the GenBank version of it. But if you click on send to file, you'll see a number of different options, including GFF3. So GFF3 is the file that allows you to add annotation to your reference genome. Okay, so here, here I added this file right here, and it allows us to see, let's remove this, so we'll go all the way out. You can start seeing that we have all of the genes of this virus, ORF1ab, spike glycoprotein, and all the rest genes right here. So this is in the GFF file. And if you click on any one of them, you can open and it will show you the actual sequence. It will show you the position start, or sorry, position start and the position end. Okay, so you can see 21,563 to 25,384. And you can see the gene lengths. So this GTF file actually shows you what is a reference in terms of the genomic element that you're looking for in your sequence. If we look into, right, so, and here's that GFF file. So if you open visually on NCBI, you can see the reference genome visually, and then you can find the GFF file down there as well. Okay, so if anyone wants this link, here's the link. Okay, I have one question. Genome-seq, which I will find on NCBI, will have COVID-19-seq and human genome sequence both will be together. No, you are just looking here at the reference for that virus, not the human genome, because what we gave you are reads that did not map onto the human genome. So you only got reference to the reads that did not map on the human genome. So we know that they're from somewhere else. And so we only map those onto COVID-19. Okay. So when we do this, right, we download this GFF file, we upload the GFF file into our visualization, okay, and we start seeing where those reads are mapped. Okay, are there any other questions about getting this done? Okay, there are no more questions. Okay, so let's talk about this just a little bit again for those of you that would like to see a repeat of this. So we have our upload, the data that I gave you, right? So the data that I gave you has two files in it. There's a reference genome. If you open the reference genome, you can see that this is the novel coronavirus. The novel coronavirus is just a sequence of nucleotides. Then the second file are 3 samples, but instead of the whole sample, we first eliminate the reads that map onto the human genome. So I already did this. I'm not asking you to do that. That's something that we will cover in the future with you so that we can study RNA expression of those cell lines. But right now, the purpose of just to try and identify whether this virus is there or not. So we depleted all the ones that map onto the reference genome, and now we take those and we want to map them onto this viral sequence. Okay, for those of you that did not see, okay, those of you that did not see these right here, right? If you had only genome genes and reference sequence or something like that, or just reference sequence, that means that you did not upload the files into the correct place. So let me again show you where each file will go. When you create the RNA-seq pipeline and you make your selection right here, you select our genome, single end, FASTQ, file upload. When you go to file upload, you have a number of different options. A lot of people just click right here on upload files. That's not what you want to do. You want to go to URL bulk upload because you have an SVL file to a number of files already provided to you, but stored on the platform. Okay, so if you have those files and you didn't get the correct output in your pipeline, try to do this now together so that you see if you got the right output. Okay, so let's go to URL bulk upload and take the SVL. Choose. Once it's uploaded, click to upload the organism. Make sure that the organism file that you're uploading is right there. Now we're not using a preloaded, we're using one from here. Okay, so there's a question, in what case can we use the upload files option? In this case, when you actually have the file on your computer. When your file is on your computer, FASTQ, SRA, FASTA, you will use add files. When I give you a reference to files stored on the server, you will use URL bulk upload or use those links right here. Does that make sense, Nikita? Okay. Now, how to check that you've done it correctly up until now? You do group selection, and you should see those FASTQ files right here. If you have a CSV file or anything like that, that's incorrect. You should see the FASTQ files right here. Okay, when you have just one link, you can use a link. When you have multiple, you use the URL bulk upload. So if we download the links, we use the first option of upload files. Yes, and I will show you here in a second, in a different example, how to do it directly from NCBI. Okay, so this was to get you started, and we'll add more complexity as we go through the program. Okay, is anyone else having any issues understanding where the different files are coming from and what we're trying to do? Or is it pretty much clear to most of you? Okay, excellent. Adekunle, we'll take a look at what exactly happened with yours. I did check your pipeline that you mentioned on the forum, and I wanted to look into it a little bit more. I didn't have time to really explore what happened there. All right, so here we don't need any groups. Okay, and then we'll do start, bow tie to G, because we're mapping on the genome, visualization, and then end. And at Nkunle, what I would also suggest is I'll show you a different example today to do something with a file from NCBI. So try that. If that doesn't work, we can even schedule a time to talk. Okay, so here this is the pipeline. You click on end, you save it, you run it, you should get the output. If you don't, let me know, post it on the forum. Let's continue so that we don't run out of time. Okay. So the second question was, where did these files come from? So how to find that? You can take these names. Did anyone find out what was the difference between these three samples? Yes, Benjamin, would you like to tell us what is the difference between the three samples? Okay, you can type it. I will just show you in the meantime, where did this data come from? So as I showed you, the actual file name is this SRR ID, sequence read archive, associated with some bioproject. You can click on all databases on NCBI and just search for it right here. All right, excellent. Thank you, Benjamin. Great answer. Let's see how to find that answer. Okay, so here you can see that this is the name of the sequence read archive, exactly what Benjamin just wrote here. Series 16, A549. A549 cell line is a cell line from lung tumors. So it is representative of the lung pathobiology somewhat. And to make sure that it is getting infected, they made it express the ACE2 receptor. But it was not infected with the actual COVID-19 virus. It was mock infection. So let's see, where do we find all of this information? Here, if you go to SRA, which is the sequence read archive, you can click on the full bio project. And the full bio project explains what exactly they did. So here you can see the explanation of what they did. They took multiple different types of cell lines. They infected them with a variety of different viruses, and they looked at gene expression. They checked whether gene expression is happening differently between different cell lines and between different respiratory causing pathogens. How many total number of samples do they have? 110. One of these 110, or three of them, are actually the samples that we gave you here. Associated with this bio project is a publication, and this publication explains in detail both what they did to generate the data and the findings from analyzing gene expression data from RNA-seq. Okay, so it's actually a very interesting study showing how the infection happens and what are some interesting factors leading to the infection, different loads of viral infection. But if you download all of the metadata from these different SRA links, you can find the three of them in here. They end with 900, 912, and 924. Okay, so let's take a look at what are the differences between them. So you can see that one of them, 924, is you have SARS-CoV-2 infected A549 cells transducted with a vector expressing human ACE2 1 hour after ruxolitinib pretreatment. Here you have SARS-CoV-2 infected A549 cells transducted with a vector expressing human ACE2. And here you have mock treated A549 cells transducted with a vector expressing human ACE2. So why do you think that one of these files did not have any reads mapped to it? Excellent. Joe, thank you for clarifying. It was a mock treatment. So, right, so SARS-CoV-2 was not supposed to be found there, so we did not find it. As opposed to these other two, they were infected with SARS-CoV-2, and so we do see them, right? Because we're just mapping them onto the genome. We're not really measuring the different elements that are being expressed from the viral genome, because we are, in this study, they're focused on the human genes. Okay. Excellent. So does anyone have any questions about how to find files and how to map them onto the reference genome, and then how to get the results and be able to interpret some of those results to understand why we find something or not find something when we map those reads to the reference database and do not find a specific virus or do find a specific virus? Did this help you understand how these different file types work together to answer such questions? Mock treated means that it does not include the actual virus. Okay, they're trying to have all of the exact same variation of environmental conditions, but not include the actual virus. Okay, to really help understand how this works, let's take another data set. Okay, Tyler is asking how are the files put into Excel. Okay, so let's take a look at how the files were put into Excel. So here we have SRA experiments. Just quickly go through this, it shouldn't take a long time. Here you have all of the different files, and you can click on send to file, and here you can have run info. Okay, we'll actually go through this again in a little bit, but I'll show you here as well. Create file. It will create a CSV file that you can open in Excel. Okay, so here it is open in Excel, and you have all of the information about these samples. Now, this includes the download links. Sometimes it includes other information that you need, sometimes it doesn't. So if you would like to actually get all the metadata, you can click on SRA Run Selector, and here we'll give you a link to download the metadata, again in the same format. Okay, so let's take a look at another project to see how we can do some additional types of analysis with this type of information. Okay, so all of you probably are familiar with the hypothesis that the viruses originated from the bats, potentially through an intermediate host. And we'll talk about the details of that hypothesis in our future lecture. But here is an example of a coronavirus in bats, a project that actually, a bio project that has a couple of different sequencing techniques that we've discussed, amplicon and RNA-seq. Okay, let's talk about what is the difference. Can anyone tell me what is the difference between amplicon sequencing and RNA-seq sequencing? What do you think? Okay, so what do you think was amplified in this case? Okay, what do you think was amplified? In this case, what were they doing here, right? So here they are talking about discovery and characterization of a novel coronavirus from a bat sample. Okay, I think Joe is on to what they were trying to do. They were trying to say, okay, could we find this specific SARS coronavirus? Let's not say SARS-CoV-2 because this was actually done in February of 2020. What they were actually trying to find was the bat coronavirus, a different version of it called RAD-G13. And RAD-G13 is a coronavirus specific to bats, but it is very close to the SARS-CoV-2. So they have one sample that was amplified, so that specific sequence was amplified. Another one is a fecal swab from this Rhinolophus affinis, which is a type of a bat, and they were trying to look for RNA-seq. What does RNA-seq stand for? Okay, what does it measure? What does RNA-seq measure? Expression. Excellent. So gene expression, right? So total RNA, or how exactly did they do it? Total RNA or not total RNA? We need to find out, but we know that it measures gene expression. But we do expect those cells to produce virus as well. So what we can do here is we can start doing the same thing, right? But instead of doing with me giving you files, we can actually do this in a different way. Okay, so here, if you think about it, taking the unmapped reads is really just an extra step, right? Because it reduces the number of reads. What if we didn't reduce the number of reads? It will be computationally more expensive. However, the genome here is not that long. It's only 30,000 genes, right? I wouldn't do this if we were mapping on a database, because the database has millions of viral sequences. But here we only have a single sequence, so we can map this sequence onto the reference genome. So let's do this together. Instead of taking some SVL file, okay, I will give you the link to the bioproject, and we can do this together, finding the data on NCBI and actually doing this from NCBI directly. So on this link, sorry, I'm going to go right here. So on this link, you will find 2 SRA experiments. Please follow along. So also you can try and reproduce this later by watching the recording. So SRA experiments, we click on this number 2. We only see 2 samples here. We go to send to, file, and here run info. Generate that file. That file will be in CSV format. Let's take a look at what's inside. Inside we have the different links, right? So these are 2 HTTP links, and they are to a compressed file on the cloud. Here we can see that this one is RNA-seq, this one is amplicon, metagenomic, this one is paired, this one is single, and so forth. You can really see how this was done, and here you can see what is the reference genome that they used for the virus. What else do we need to be able to map this onto the reference genome? What else do we need to map this onto the reference genome? The reference genome. Excellent. How do we find the reference genome? The reference genome is again on NCBI. Okay, so we're going to, I have a few things open here. I'm just going to close them. Okay, so here, if you look for rat G13, and instead of SRA, we can just do all databases. So let's search for that genome, the reference genome. And here is the, okay, bad coronavirus, rat G13. So this is the actual genome. So to download this reference genome, what we need to do is send to, file, and then here you select FASTA. Click on create file, and you will have a FASTA sequence. If you open the FASTA sequence, you can see that it is the bad coronavirus RADG13 complete genome. So once that is done, now we're ready to make our pipeline. We'll use single end. It doesn't actually matter. We have two files. One is paired end, one is single end. It doesn't matter in our case because we're just mapping them onto the reference genome. We're not actually counting RNA expression right now. So here we'll use our genome, single end, FASTQ, file upload. Instead of uploading this button, you see that this option is NCBI run table upload. So we'll use this button right here, add files. Okay, those were right here. So we'll upload that SRA run info file. Then we'll click on organism and we'll actually upload here the FASTA file that we downloaded. Once you click on group selection, what's important is to actually see here the SRA files, not the CSV links, but the actual SRA files. That means that the system has processed those links and identified those two SRA files. Cancel, no groups, start, bow tie 2G, visualization, end. After this pipeline is completed, this is what you should see. How to check what did you actually upload and whether everything uploaded correctly. You can go to uploaded data and you should see not only the NCBI, but also the actual extracted files. And you'll see that they'll come in three formats: SRA, FASTQ, and .fq. .fq and .fastq are the same thing, but they're still being converted because some algorithms deal with only FQ, some deal with only FASTQ. Okay, so if this has actual files and you can see that they have sizes, that means that you've set up your pipeline correctly. What are the steps? Start, bow tie to G, visualization, and end. As a result, you should see a couple of things. One thing we'll look at are the mapping stats table, and the other one is visualization. Let's take a look at the visualization first. In visualization, again, I can upload a reference to help me see the different genes in the reference genome. The second thing I should see is coverage, like we saw in previous one, and alignment. Alignment is of actual reads, and coverage is total reads per position. What do we see here? This is, right, this is one sample, 578, and it has a total of just 4 reads in this maximum position. The second one is 797, and it has more, right? So up to about 40 in this sequence. Which one do you think is which? Which one is the amplicon and which one is RNA-seq? So we have two samples here, 578 and 797. Which one do you think is the amplicon and which one is RNA-seq? Okay, Benjamin is saying that the top is amplicon, 97 is RNA-seq, second is amplicon. Okay, amplicon is 578. Okay, so again, think about it. You can either think about it or you can actually go back to the bioproject link that I gave you and take a look there, right? So let's take a look. Which one is, so we can actually see it right here. $797 is the RNA-seq, and 578 is amplicon. Okay, so let's do, so 578 is the amplicon, and 797 is RNA-seq. So RNA-seq is expected to generate a lot more data, right? A lot more reads, but a lot of those should fall onto the reference genome from the human. Let's take a look at what other outputs do we have to help us understand the overall number of reads mapped onto the reference genome. To do that, we can download this file right here, mapping stats table. Once you download this link, okay, this is the mapping stats table. It will come in as a TXT file, but you can actually open it in Excel. So if you open it in Excel or any kind of table format, this is what you will find inside. This is a little bit cleaned up. It's a technical file that is a little bit difficult to structure and directly view. So I cleaned up a few characters here. Again, remember that we have two different ones. This is the RNA-seq. This is the amplicon. Okay, let's. How many total reads were in the amplicon? Only 33 reads. 100 of those were unpaired, aligned 0 times 0, 33 aligned exactly 1 time, 0 aligned 1 time, 100% overall alignment rate, right? So all of the reads that were present in this specific file were from this RAD-G13 virus, viral sequence. So just mapped onto the reference genome. However, here you can see that it's 0.19% overall alignment rate, right? So the majority of the sample is actually from the host, not from this virus. But even though we have such a small number of reads that aligns to the viral genome, we actually have 13,689 reads that mapped onto the genome one time, and then another 31,000 that aligned more than one time, right? So which means that in total you have about 13, 14,000 reads compared to 33 reads, right? So very small number of reads, a lot of reads. And even in the small proportion of reads that actually mapped onto this genome, we do see a fairly nice coverage of the genome, right? So here you can see coverage. Okay, any questions about what we did here so far? All right, so here we can zoom in to see the actual sequence, right? On top here, we see the sequence. Again, I don't know if we need the reverse strand and translation right now. Let's just keep it simple. And let's take a look at what information we can actually see inside this data set. What else do we see beyond just the coverage? Does anything else pop out? Okay, SNPs. Excellent, thank you. Okay, so let's address first some of these questions. So simply seeing graph, I can't say which graph belongs to RNA and which one amplicon. Right, you have no idea by just looking at the visual picture what this is until you dig into the data and try to understand where did this data come from. The other question is, just to clarify, the data we got from RNA-seq is significant for presence of virus based on number of reads and not the percentage. Okay, yes, and we'll talk about that. That is a part of the SF9 project. We'll take a look at some examples to see, okay, do we know that this is actually the full genome present here, or just some random portion of the genome? As we saw in the previous example with the cell lines, you get over 90,000 reads at the end. Not only that, you also get, right, if we take a look at, let's take a look here again. So here, right, we have 3 different samples. We know for sure that this sample does not contain the virus. Did any reads map onto this sample that does not contain any of the virus? Yes, some of the reads mapped right here. Does it mean that the virus was present? No. How do we know that it wasn't? Well, look at the difference here. You have 90,000 compared to about 2,000 here in the middle. And here you have 4 compared to 0 right here, right? So there's a lot of mapping quality issues that come up from amplification and library preparation techniques that also play a role in what we can actually identify. So if we see just some random section covered with a number of reads, that does not mean that the whole virus is there, right? If the main portions of the genome are not present, that means that the virus is not present. Okay, how do you add the GTF file downloaded into the visualization? Great question. Let me show you. So bad coronavirus visualization, click on visualization, it opens up. And here you have just the samples and the reference sequence. You can go to File, Open, and here select or drag files, or you can even paste the remote URL. I prepared these files for you, but I already showed you how to find them. So let me show you. I will actually share this with you after the session is over. Okay, here I have the BAT RAD-G13 GTF. So you just drag and drop it right here and select here that this is GFF3. Open. Okay. And what happened here? Let's reload this again. File, open. Okay. Something here is wrong. Maybe I'm using the wrong data set. Okay, important to actually, let me see. So I believe it's this one. Visualization. Okay, there you go. Something was wrong there. Genome genes, let's do open. There we go. Okay, so you just add it right there, and there we have all of those genes. Okay, so I do have a number of other things that I want to just quickly cover in here. So let's, I know there's a lot of questions of about the visualization and things like that. So I'll share some links that you can take a look at. JBrowse settings. There's a lot of different settings that you can actually change on JBrowse and kind of adapt it to whatever you're trying to do. But the purpose here is to focus on trying to see the differences in coverage and seeing what else can we discover in this data, okay? Okay, so we saw how to load the reference sequences. We got the visualization results. What else is available? Let's zoom in a little bit. And we start seeing these little symbols underneath of each track, right? So here we see those SNPs or variants. So we actually see highlighted different positions on this specific sequence where there's an alignment between all of the reads present in some variant compared to the reference genome. Whenever this variation is in majority of the reads, see here you have 3 reads, 2 of them have a variant and 1 does not, and you see the variation highlighted. Okay, so that is what these little dots or symbols will have. Okay, whenever it's just 1 read, it's not going to have that dot. So you can scroll through the whole genome and start seeing how those variants appear in different reads. Now, the number of reads here starts playing a major role, right? Because when we look at the specific sequence and we try to see in that sequence how the sequence is related to the reference genome, just like we're seeing here, we need some evidence about the differences, right? Each difference that we find needs to be supported by some number of data points that we'll have to prove that this is a real variant. Okay, so let's pause. I'm going to add another few minutes at the end for the questions. I do recommend going through the specific example in SF9 that will show you how some of the things that you were asking in today's session actually play out in a real situation, right? So here we kind of are dealing with a simplified situation. The simplified situation is that we are taking files that were prepared in a lab or that were sequenced in such a way that they already knew what they had, right? It was not a discovery situation. It was not like taking some sample and trying to discover whether it has something or does not have something. So in a real situation, which is the SF9 project, they actually took some commercially available cell lines, sequenced everything that was in there, started looking for specific, started looking for all the possible viruses that they could find. How did they do it? They did it actually in a different section, right, that you will see a lot more described in the SF9 project, under metagenomics. Here we only were mapping onto a single genome, right? That single genome contains specific sequences, and if we don't find something, we don't know what else is in there. But in here, instead of one single genome, we can actually use a whole database. So for example, one of the options is to select a virus genomes database. This database contains a lot of different types of sequences. Here you can see 219,000 sequences just from one type of a virus. If you have your microphone on, please turn it off. Okay. So all of these sequences are very different. And so when we are interested to find what is in our total RNA-seq data, we can map onto this reference database. It will be a much longer process because this process is repeated hundreds of thousands of times. But at the end, what you will have as an output is a list of sequences that have some reads mapped onto them. In the SF9 example that you can go through in that project, you'll see that a lot of them might have some reference, some importance for what you're looking for. For example, here you can see different types of viruses that were found in a specific sample and the number of reads associated with that one. What are those viruses? Here's an overview of some of those viruses. It includes SF9, the rhabdovirus. It includes simian virus 40, okay? Simian virus 40 is SIV, kind of like HIV, but only for primates. And a bunch of other viruses like hepatitis C. Now imagine if this was actual viruses present in a cell line that was used for production of biologics, for production of insulin, for production of vaccines, etc. There's going to be a huge contamination issue from the from the FDA perspective, if this cell line is producing all of these viruses. So SV40, in fact, was suspected to be associated with a polio vaccine. So you can see here that vaccines used in the late 1950s and early 1960s were contaminated with a virus called Pseudovirus 40, present in monkey kidney cells used to grow the vaccine. And then they even found those viruses in patients with cancers. And there's a big controversy about suspected viruses entering that process. So how do we know whether just the number of reads present in, mapping on these references plays any role in the whole virus actually being detected? If we map the reads, this is the reads taken from that whole project and mapped onto SV40 reference project. You can see they only map, there's a lot of them, there's like 15,000 of them or something like that, but they only map to a very short segment right here, and they are just stacked. And the problem is that this virus is obviously a genome of close to 5,000 nucleotides long, and here we can see a very small portion stacked up together. And this typically represents some kind of a technical issue when reads are nicely stacked up, just like what you're seeing here, and not spread over a wider portion, which would mean actual biological presence of that organism in a sample. So for us to really validate some of these findings, we have to make sure that we go through the full pipeline. So sometimes we will map onto the reference genome to eliminate the reads that map onto the host. We can then map onto the reference database from the viral sequences. To validate, we need to map on a specific virus that we are interested in identifying. And that includes validation of how well those reads and how many of those reads align to the full sequence. And then potentially also performing assembly to make sure that if it's a novel variant of the same virus, for example, when they discovered the novel coronavirus, they didn't have a reference yet. They had to use some SARS-1 or other bat viruses as a reference initially, and only then assemble all the reads that we have into some kind of a variant of that same genome. Okay, so has anyone looked into the SF9 project? Okay, great. So please take a look at the project, see what's in there, try to replicate some of the steps, see if you understand how the mapping results could be interpreted, and try to think of how the same principles could be applied to the discovery of the novel coronavirus, for example. Okay. Okay, so if there are any questions on what we covered today, I want to briefly introduce the topic that we will go on next, which will be after we find what the reads map onto and have a sense of how they map. Okay, Therese is asking, is there a general threshold you can use to determine that a certain number of reads is negligible, or should you look at it comparatively with other samples? Depends for what, right? And that's a great question. So we started talking about some of these, so to say, variants, okay? So one of the questions about variants is whether it is a variant that we are sure of or a variant that we are unsure of. And we'll talk in our next session about multiple sequence alignment. And of course, when we use multiple sequence alignment, we use that genome sequence without the sense of how many reads map to which place. So we just have sequences of either nucleotides or amino acids even that we compare to each other. That gives us a lot of biological interpretation capacity, but it doesn't provide us with a good sense of where that data originated from. As we saw in these examples, in reality, we deal with something that looks like this. we have a number of reads, which is called coverage. We have a reference sequence where we have the nucleotides in each position on the reference genome. And we're trying to look for variants. And those are going to be different types of variants: substitutions, insertions, and deletions, different types of variants. So when we have a situation like this, where we have 1, 2, 3, 4, 5 reads that all contain a change from the reference, right? We're going to be fairly certain that this is a substitution, and we can mark it as a substitution. But what if we have only 2 reads? What's the problem with 2 reads? They can have conflicting evidence. They can have one is saying this, another one is saying this. There's no third one to really break the controversy, right? So we need at least one other one to know which variant here is the actual substitution. In other cases, you'll see insertions and deletions. What kind of evidence is needed to identify insertion and deletion? At least a couple of reads that show you that both of them have a continuous sequence based on what we see here, right? Certain nucleotides are missing. So obviously this is an artificial example, but it shows you how important it is to have a significant coverage. And for example, for whole genome sequencing, the rule of thumb, if you're interested, is 5 reads per base. So you need to have at least 5 to make a determination that is somewhat significant in terms of a DNA reference genome. In RNA genomes, the situation is even more complicated because RNA viruses have a high mutation rate. And we don't have the pair strand to really know whether that was in both strands or no. So it is more complicated. And we'll talk in other programs that we have, in the program that we have on bioinformatics for infectious diseases. We don't have that session scheduled here, but we can talk about these methods of ultra deep sequencing that help us discover low frequency variants. Because not only does the sample represent a collection of cells, so really many different viruses together, but also represents various types of mutations that it is actually impossible to clearly characterize just using next generation sequencing. So depth is very important. That's why you can see 2,000 reads mapping on those specific viruses. When we characterize viruses and want to understand all of the different mutations, we really need to pay attention to how many reads we have. Okay, so here, right, when we look at the number of variants present, they will have different confidence levels. Some of them that appear across all of the reads consistently, we are fairly sure that that's what they are. Some of them are really probably mistakes or rare variants. Again, remember that the sample typically is a number, huge number of genomes actually present. Okay, can you explain the process of identifying a novel virus? Is there a threshold in variation to designate the virus as novel? Well, yeah, it needs to have specific changes from what is known. But I think that, in our next few sessions, you'll see a lot more about how different genomic sequences compare and what type of information we can extract from different types of data sets available, which will give you a sense of what a novel really means and how it compares to other known viruses of similar species or other hosts that contain those viruses. Okay, are there any other questions? We're going to be wrapping up here. All right, thank you, Manish. Okay, so in our next session, like I mentioned, we are going to be going through multiple sequence alignment. Multiple sequence alignment is the alignment of genomic sequences to each other, and they could be either whole genomes or individual portions of the genome. You can use SRA files that we've used up until now and potentially read coverage of those genomes in multiple sequence alignment as well. So there's a whole variety number of options. We can use multiple sequence alignment to study variation, how closely related different genomes are. And specifically with the novel coronavirus, that's a great point to start our exploration into the origins and the potential sources or reservoirs of this novel coronavirus. Which organisms, like bats, we saw today, we saw that they contain some closely related novel coronavirus sequence. How closely is it related? How closely or how different are those reservoirs compared to what we find in humans, right? So coronaviruses have been around for a while. So all of these things we'll talk about in our next session. Again, just like we did last time, I'm going to ask you to try and replicate the analysis that we did today. To do that analysis, what you are going to need are the two samples, right? So we have these two samples. You need the reference genome, you need the GFF file, and then you'll need to generate the same example visualization like we did last time. We'll give you some ability to practice and start looking for specific positions that you think are different from the reference genome. So I will, again, on the forum, share with you the files and the directions to follow. If you have any questions, please review this video. Or if you're not getting the results that I am getting and showing you in this session, then you can put those on the forum as well. We'll make sure to address those and check your pipelines individually. Thank you for joining today. The video recording will be posted on the forum shortly, and I'm going to be looking forward to seeing you again next time. Thank you.